Hi everyone, my name is David Wood, and I'm here with my co-founder and CEO, Izabela Gierach, to tell you about our new company, Protein Capture Science. We have a technology that we believe is going to revolutionize how biopharmaceuticals are discovered, developed, and ultimately manufactured. To tell you a little bit more about the technology, I need to explain a little bit about how biopharmaceuticals are made. And so typically, these processes are divided into upstream and downstream processing. In upstream processing, a piece of usually human DNA is put into some kind of a cell that can be grown in a big tank. That cell makes the human protein, and then in downstream processing, that protein is purified to a point where it's safe to put into a human. The difficulty, the problem that we've addressed, is that this downstream purification can be very difficult to develop. It costs a lot of money, and there are huge yield losses depending on how the purification is developed. One of the platforms that, because it's so powerful, has become so incredibly universal is what they call the protein A affinity platform. In this case, there's a single column that you can think of like a Brita water filter that very selectively grabs monoclonal antibodies out of these mixtures of cells expressing the protein. Those monoclonal antibodies become the drug that's eventually injected into the patient. And protein A allows you to go from a complex mixture in the crude feed to a very highly purified protein in a single step. The question is, what about proteins that aren't monoclonal antibodies? And this represents about 3/4 of all of the molecules out there on the market. Those proteins don't have much in common, and as a result, you have to develop complicated purification systems where each step removes a small subunit of the contaminating proteins. And as a result, these processes are hard to develop, they have large yield losses, and they usually represent a large majority of the cost of making the drug. So the question is, how can you purify all of these other proteins using a single Brita water filter column the way you do with monoclonal antibodies? Now, in research, this problem's been solved by adding what they call an affinity tag or a molecular tag to the protein that allows you to purify that protein with a single step. The difficulty is that even though you can use that tag to purify all these different proteins, once you have that protein purified, the tag is not human and may cause an immune response in your patient. And the last thing you want is your patients becoming allergic to your drugs. So the question is, can we develop a new single platform that's based on a tag, but the proteins coming out of that platform have no tag on them? This would be unique and uniquely powerful because it could be used both at research during discovery, but then the exact same process could be used in manufacturing. And it de-risks and streamlines the entire research and development program. So the key to this technology is that it allows the research and development, which depends on tags, to be bridged to manufacturing under a single platform. Normally, when you transition from research to manufacturing, there's a lot of risk involved. But if you can use a single method to do both, then you reduce the risk, you streamline, you decrease the cost, and you accelerate these drugs to market. And this technology that we've developed is unique in its ability to do this. So the way the technology works is we have a protein called an intine that is split into two pieces. One half we attach to our Brita water filter, and the other half forms the fishhook, the tag that we put on the target protein. When you take that tagged target protein and you pass it over that Brita water filter, the Brita water filter grabs onto that tag very tightly, and when those two pieces assemble, they form a self-cleaving ability where they can release the target protein while the tag stays on the resin, thus delivering something that can be perhaps even injected directly into a patient. So in 4 easy steps, we've solved the problem of purifying a protein quickly and easily, and we've created a platform that will work with any protein, even if it's not a monoclonal antibody. We've been very careful to make sure that everything that goes into this product and its design is the boxes that companies want checked for these types of resins and these types of technologies. And we've also worked with partners who can scale these processes up so that they can easily go from laboratory scale into manufacturing scale. And we've produced many case studies and examples, including filgrastim, which is a biosimilar of Neupogen, and the COVID-19 spike receptor protein, and shown that this single platform can easily purify both of these two targets. And so that's the technology that I wanted to show you. And going forward, what I want to do now is hand off to Izabela, who can tell you about the market opportunities and where this technology is going commercially.
So the protein purification market, it's an $8 billion global market. The subsection of it for affinity resin is about 1 billion. And that's the market which we wanted to go after. Obviously, we cannot do it with the best team. This company was created by 2 PhDs. David Wood, which you just saw, he developed this innovative technology, and he graduated from Caltech as well as RPI. He also worked at Amgen and BMS. I have a PhD in biochemistry. I graduated from University of the Sciences in Philadelphia, known for the first pharmacies college here in North America. And I also have an MBA, and I worked previously on products development and diagnostics development. We have a great support from Rev1 Venture, and additional people who are really close to our company are Woody Hassel and Christy Melnick. Current status of this product is that we have 4 issued patents, which we licensed from OSU. The 5th one is on the way. We also have a trademark, which is very important for us, of the name of ICAP tag. We were able to develop a small scale of non-GMP material, and we would want it in the future go to the GMP level products so they can be used in the biopharmaceutical setup. We wanted to connect with the customers, expand our channels of distribution, and bring more marketing material so everybody globally will understand what we are doing here in Ohio. So you can see more information on our website, www.proteincapturescience.com. You can also see us on the podcast, Apple Podcasts, as well as through the JohnScript webinars. We are here to change biotechnology as we know it, and I hope you will join us.
We have received a question on your regulatory strategy and timeline.
We've identified a few early adopters who are beta testing our material now. We have even some paid sales from large companies who make biopharmaceuticals. So in terms of creating a product that would be used in manufacturing, our product would become part of what's called the CMC package, the Chemical and Manufacturing Controls Package. And it would just have to be very, very well documented. And we've talked to a couple of manufacturers who are willing to make the sort of GMP grade material. As you can imagine, it's a little bit expensive because they have to make a fairly large scale manufacturing run 3 times and show that it can be done consistently and well. And so what we've focused on now is identifying partners throughout everything from academic basic research clear to sort of Amgen level companies who would be interested in the technology and letting them try it out. So we're sort of in the customer validation stage. We've completely developed a great manufacturing process, and we've talked to companies who are willing to evaluate that process in a GMP context. But for right now, we're focusing on getting it out there and getting people excited about the technology. But the path is clear to GMP grade manufactured resin. And there are already companies who are asking us for quotes on much, much larger volumes.
Interesting. So you mentioned you're in that customer validation stage. What do your current customers look like, and how are they using your technology today?
So we've sent the technology, either paid or free samples, to several different types